Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Aviras, and in this video we will be discussing about protein synthesis. So DNA acts like an instructional manual for protein synthesis. It provides all the information needed to function. The actual work of translating the information into a medium that can be used directly by the cell is done by the RNA. Okay, now RNA is which is also called as a ribonucleic acid. So RNA is a similar molecule to DNA, and it is made up of a long chain of nucleotides. So there are three main differences between DNA and RNA. Single-stranded, sugar is ribose, and in DNA it is deoxyribose. It contains uracil and thymine group. Uracil group instead of the thymine group. So the types of RNA. So one is the messenger RNA, which carry instructions for making proteins, and it is made during transcription process. rRNA or ribosomal RNA is along with proteins make up the ribosomes. And number three is the transfer RNA, which bring the amino acids to the ribosome to construct the proteins. So reading the DNA code. So what is the DNA used for? So DNA is used to make proteins that control the growth and repair of in a living organism. The steps to making a protein synthesis, is from DNA to RNA to protein. The process from DNA to RNA is called as transcription, where we change the writing, and from RNA to protein is the translation, where we change the language, we can say. So RNA is a disposable copy of a part of DNA, a working copy of a single gene, and its main job is to be used in protein synthesis. Now, why can't we just send DNA out into the cytoplasm? So because it will get damaged and information could be lost. So that's why we send RNA in the cytoplasm for protein synthesis. So transcription is a part of DNA molecule is copied into a single strand of RNA where we change the writing. And where does it takes place? It takes place inside the nucleus of the cell. So the transcription factors are the proteins that bind to the promoter regions of the gene. They show the RNA polymerase where to bind. Now, what is RNA polymerase? Now, it binds to the DNA and it separates the strands, and it uses one strand of the DNA as a template to make the RNA. Now, step one is initiation of RNA polymerase binds to the DNA promoter region. Now, what is a promoter region? So it is a specific base sequence that RNA polymerase binds to. The promoter contains an initiation site where transcription of the gene begins. Now you can see this is the promoter region. Transcription factor binds to the Tata box containing the TATA sequence, and then the RNA polymerase binds here, the transcription factors bind here, and then the transcription initiation complex is formed. So step two is the elongation. That is, the nucleotides are bound together based on the DNA template, and it assembles the RNA strand based on the template from 3' to 5' position. Okay, the direction is from 3' to 5'. Now, this is very, very important. So now here you can see this is the 3' position, okay, and this is the 5' position. The transcription of the antisense strand is occurring, okay, and this is the 5' position, T with A with U, like that, and this, and again then it goes to as a translation of mRNA formation. Step two is RNA polymerase arranges the nucleotides that are complementary to the DNA strand being copied. RNA contains uracil instead of thymine. A gene can be transcribed many times by multiple RNA polymerase molecules. Step three is the termination. That is, the RNA polymerase stops the transcription. The RNA polymerase continues to elongate until it reaches the terminator. So what is the terminator? So termination is a specific sequence of nucleotides that signals the end of transcription. RNA editing. Where on the DNA strand does transcription begin? At the promoter region. The extra DNA is transcribed into the RNA. So DNA contains nucleotides that are not involved in making proteins or introns. Before RNA leaves the nucleus, the introns are cut out, leaving just exons, sequences that code for proteins. Now again, then there is the translation. So the mRNA leaves the nucleus and is used to create a protein needed by the cell. So now RNA is converted into the proteins, where we change the language. So proteins is made up of a long chain of amino acids called a polypeptide. So 20 different amino acids are present in the human body, and the code for making proteins is found on the mRNA strand, that is the messenger RNA strand. So the genetic code, the mRNA strand, is read 3 letters at a time. Each 3-letter sequence is called a codon. Now, some amino acids are made by more than one codon. So there is a start codon and there is a stop codon. So start codon is a 3-letter word which indicates the start of the protein, and stop codon, where it ends the translation, does not code for an amino acid. Okay, so one codon equals to one amino acid. Translation takes place in the ribosome, and it has 4 steps. First step is, as the mRNA strand is read by the ribosome, tRNA begins the tRNA brings the correct amino acid. Now, each tRNA carries only one amino acid, and tRNA has three unpaired bases, which is called as anticodon, that are complementary to the mRNA strand. Now, what we know is that in mRNA, there is a start codon. Okay, for example, this is a codon GCA. Then another anticodon will be there, which will be binding to these. Okay, and that will be brought in by the tRNA. Right now, the amino acid is transferred to the growing polypeptide chain, and each ribosome has two binding sites. The ribosome adds a polypeptide bond between the amino acids, and then the translation continues until a stop codon is reached. Now, the name of this stop codons is very, very important. That is UAA, UAG, and UGA. Remember these three names by heart. So putting it all together, a short length of DNA is copied into mRNA. mRNA leaves the nucleus and enters the cytoplasm. A ribosome reads the strand of mRNA in sets of three and matches the tRNA with its amino acid. The amino acids are released from tRNA and bound together in order making a polypeptide. The polypeptide folds into functional protein to be used by the organism. Thank you for watching. Keep studying.